Yeah, so let's start the morning session. So yesterday morning, you heard from Anshul, and Surag is Anshul's PhD student at Stanford University. He did his undergrad in electrical engineering at IIT Delhi, and he's been working on neural networks, deep learning, and regulatory genomics. And yeah, over to you, Surag.
Thanks, Rahul. Hi, everyone. Thanks for showing up this morning. Thanks, Rahul, for organizing and the other organizers as well. So I'm Surag. I'm a PhD student in Anshul's lab at Stanford. And we do a lot of work on, as Anshul mentioned to you guys yesterday, DNA sequence models. So these are models that take in DNA sequence of different lengths, can be as short as 500 bases, can be as long as a mega base. So 1 million bases as input, and try to predict different aspects of cellular biology that are predictive just from DNA sequence alone. And so I'll be giving you a flavor of that today. In fact, later today, we have a hands-on session where we'll try to train models in Keras, which is in TensorFlow, sort of build models from the ground up. And we'll see that these models, without telling them anything about what features are present in the sequence, and end up learning the correct features and end up learning the correct patterns that are necessary for making accurate predictions. So just to set up, I know Anshul gave you a talk yesterday, but I think there's a few aspects that are yet to cover that'll be relevant for the tutorial later today. And so I'll be quickly going over some of those. And the goal of my talk is not to give you sort of a lot of technical jargon and just a lot of concepts, but to get you to be able to provide you with an intuitive sense of what's really encoded in the DNA, what makes a cell, right? Why are different cell types in our body that share the same genome different from each other? And so once you get an intuitive sense, we'll be able to translate that to how we design models for regulatory DNA, right? So you've seen the slide yesterday. So the DNA really is this really one big long string, right? In humans, it's about 3 billion bases long. So each of these characters is a base, right? And we have 4 bases, A, C, G, and T. And the genome really is 3 billion bases long. And it's crazy because it's not a language that we can read as humans. So unlike English or Hindi or Marathi, these aren't languages that we've designed. And so these aren't really languages you can read left to right. In fact, there is no notion of left to right here. You can actually read it equivalently from right to left. So what people have been working, for the last century and beyond that is figuring out whether it's the entire 3 billion bases that hold meaning, that are relevant, or if it's subsets of these 3 million bases. How do these 3 billion bases interact with each other? Are they completely independent? If I make a change here, does that affect the function only if that's relevant within this? Or perhaps does it have effects that are linked to another piece of the genome that might be like 1 million bases away, right? So these are things people have been thinking about and trying to decode. And as many of you might know, about 2% of our genome is what's known as the coding genome. And this is the genome that codes for proteins. So this is the genome that's transcribed and then translated into proteins. Proteins, as you know, are these molecular machines that are relevant for for carrying out different functions within our cells, right? And so often, if you do have mutations in regions of the genome that code for protein coding sequences, the consequences of that can be pretty severe and can lead to different kinds of genetic abnormalities, right? But often what we see in most healthy individuals, right, most of us sitting here, is that for the most part, our protein coding genome is intact, right? It's pretty safe. It's not deleterious or harmful in any way, and that's what allows us to sort of grow up and be fairly healthy. However, there are mutations in what's called the non-coding genome, and that's genome that's not this 2% of the genome, right? So you can really think of proteins, and feel free to keep asking questions. Let's keep this interactive, and we don't have to cover all the slides even, just to give you, the idea is to give you an intuitive sense. So please stop me if you have questions. And so you can think of the protein coding part really as the ingredients of your genome, right? And the rest of the genome as, okay, these are loose analogies, but I think it helps me definitely think about this a lot more clearly. And you can think of the remaining 98% of the genome as really the recipe, right? And so you can imagine if you're making a cake, you have butter, you have eggs. If you mess up the ingredients, that's not really going to help you build a successful cake, right? If your eggs have a defect in them, if your butter is not butter, it's completely it's gone bad, it's going to have issues. However, if your ingredients change in different ways, you might still end up with a successful cake, right? For example, if you just make spelling errors in your recipe, right? Perhaps it's not a big deal because the person making the cake can figure out that there's a spelling error and fix the spelling error. Right? However, if you interchange steps in that recipe, then again, it can be disastrous, right? So that's how I like thinking about the genome in general, as with the protein coding elements being the ingredients and everything else really being the controlling elements that tell you how to combine these ingredients in meaningful ways to give rise to cells, different cell types, tissues, organs, and the entire organism, right? So yeah, as Anshul told you, all our genomes, all our cells contain the same copy of our genome, right? So the same 3 billion bases times 2 maternal and paternal copies. And so all our cells, each cell in our body, for barring some exceptions, harbor the same book, right? So this DNA is this big book, and every cell in your body has a copy of the same book, and yet different cell types are very distinct from each other, right? So neurons are very different from heart cells. They have different functions, different morphologies, different division times. Some don't divide, like neurons are terminal, they don't divide. Some cells divide a lot, right? So a lot of these properties, despite having the same book, are very different among different cells, right? And so the way I like to think about it is that different cells are reading different chapters of this big book, right? And there's actually ways to measure that. And I'll talk about that in the next few slides. So you can think about the brain reading brain-specific chapters, right? Neurons reading neuron-specific chapters of the book, and really it's closed the remaining books, the remaining chapters of the book. The heart's reading heart-specific chapters of the book. But also all cells and different cells will read some common like metabolism-related and housekeeping kind of chapters, right? So there's definitely some commonalities in terms of the metabolism and basic cellular functions that's shared across cells. And so all cells will read sort of that shared part, but then they'll also read specific parts that are specific to their own function, right? Does that make sense? And that's what really leads to this diversity. Is basically determined by how different cells read the same genome, but different parts of it, right? And there's actually ways to measure it. And often these bits of information, so of course the protein here are the key ingredients, but there are portions along the genome that are called regulatory elements. And these are, you can think of those parts as the ones that are really being read by that cell, by a specific cell. And these elements can be different in different cell types. And there's some interesting sort of biochemical activity at these elements that I'll talk to you about. But these really are the control elements. So you can think of this as being a specific cell type, let's say a liver cell, and these as being liver-specific control elements. And so now if you change these elements, if you sort of mutate the sequences in them, it's not that the liver will likely stop functioning, but then the function will be altered in subtle ways. Maybe the expression of this gene, the amount of protein produced in the liver cell will go down by 10%, 20%. And maybe that might not have severe impact on the function of the liver as such, but perhaps like cumulatively over 10, 20 years, it might lead to more complex diseases, right? So people have been able to perform a lot of biochemical experiments that figure out what parts of a cell are being read. And just to give you a sense, you can think of the most interesting one being something called chromatin accessibility. And this is a physical meaning. So in fact, what this means is that there are regions of the genome that are compacted and literally they're closed and not available to be read, right? And so often there are experiments, things like ATAC-seq, DNA-seq, if you've heard of them, that allow us to figure out what regions of the genome in a specific cell type or tissue are being read. And so if you look at this, so this is actually the genome on the x-axis. You can imagine this as being perhaps like a 1 megabase slice of the genome. And you'll find that there are regions within the genome that are accessible. And so these are actually physically open regions that have proteins going in and out, and that are sort of linking to genes and sort of driving expression of those genes. And these really are the functional paths for that cell. And this part can change from these marks can really be different across cell types and are reflective of what paths are being read, what part of the genome is being accessed in that specific cell type, right? And so in addition to accessibility, we also have things like where certain histone marks are present, if you're aware of those. But these are essentially certain proteins that decorate the genome in many ways. And so let's say this is a specific protein. What this is telling us is that along the genome, there's only specific parts of the genome where you can find this specific protein, right? And so proteins also come in different flavors. There are transcription factors, which are proteins that actually bind the DNA. They recognize certain DNA words that I'll talk about, and drive about sort of downstream function, right? And so, for example, this can be another transcription factor, and you can see that it's binding in regions that are open, but it's only binding a subset of regions that are open, right? So you can see a lot of this combinatorial behavior where there can be many regions of the genome in a specific cell type that are open and accessed. But if you look at it from the perspective of a single protein, from a single transcription factor, it might only bind to a subset of all open regions. That will be determined by the underlying DNA sequence itself. And so, over the last few decades, people have collected this compendia of data. Really, you go across cell types, you go across different experiment types, and people have been able to collect thousands of experimental samples that map these proteins and regions that are accessible across hundreds of tissue types and cells. And in fact, more recently, there's been work on expanding these collections, not just to entire tissues, but to look at this at the resolution of single cells. There's been a boom in single cell technologies that allow you to profile exactly these properties of the cell, but at single cell resolution. And so you can imagine, like, before, what used to happen was people would blend up entire tissues, and so you'd get an aggregate readout of what's being read in this organ, right? And that organ can be is usually composed of multiple cell types in different proportions. But what's happening now is that you can really figure out within cell types, within an organ, what are the regions that are accessible, what are the regions that are being read. And so that's really opening up this new wave of analyses that are powered. And a lot of this data is publicly available. If you have heard of the Geo Archive, most sequencing data that people collect is deposited on this website, and you can actually go and access this completely for free. And there's literally terabytes, petabytes even of data, sequencing data that's just sitting there and something you can access at the click of a button. Just to let you know. So the theme of the this talk, and also the tutorial late in the afternoon, is to learn predictive representations of input sequence from any given data set, really, and to try to figure out if you can learn predictive features without telling the model what the predictive features are. So we're learning this de novo, right, without even telling the model, as opposed to sort of old machine learning methods where we had to hand engineer features, right? So you tell the model perhaps that GATA is a specific DNA word that you should take account of, right? Or something that comes from pre-existing knowledge from literature. But now with the advent of sort of machine learning and deep learning methods, we don't really have to specify these features based on our prior knowledge. We can completely learn these in a data-driven way. And once we've trained these models, we can also interpret these models to see what these models are learning, whether they're learning the right biologically meaningful DNA words. And so we'll cover some of those aspects. So let's think about regulatory sequence a bit more and how proteins called transcription factors act at these regulatory elements. So it'll be helpful to think about all of this in the context of specific cell types. So let's imagine we're looking at a liver cell, right, hepatocyte. And so what can happen is often at these regulatory elements, as I told you, these are physically accessible. So these are not compacted. So really these are open parts of the genome that can be accessed, like proteins can shuttle in and shuttle out, access these regions, and then do something else downstream. So there's relevant function. Just by binding at these sites, these proteins can bring about some relevant downstream function. So as I told you, these transcription factors often recognize within these regions of open DNA certain DNA words, and this DNA word is often called a motif, and it's specific to different transcription factors. So humans have about 1,600 transcription factors, and most mammals have about the same number of transcription factors. But it doesn't mean that there's 1,600 unique DNA motifs. A lot of these transcription factors are related to each other, so they're basically homologs and families of transcription factors that have very similar motifs. So really, effectively, you can think about there as being 100 to 200 different unique DNA words in the genome, right? And so a transcription factor will probably recognize a DNA word that looks like this. So let's assume there's a transcription factor that prefers binding this DNA word. Let me just tell you how to read this DNA word in the first place, right? So it's really not like an English, it's very much like an English word with some subtle differences. What this is telling you is that this transcription factor will physically bind to DNA that contains this sequence, right? This A-rich, G-rich sequence. But you can see that in many positions like this one, the height of the A is different from the height of the A here, right? What that means is that this specific transcription factor doesn't necessarily need to see an A here. Like it likes to see A here, but it's okay if it sees a G here. So these are soft words in some sense. You can think of them as, like it lets you generate all possible strings of this length. It's not that the transcription factor will only prefer one exact match, string match, right? It'll prefer multiple string matches with different binding affinities. The ones they'll prefer the most will probably be the A, G, C, G, G, A. So the most common word, common base at each position. But even if you have a sequence that has, say, a G here or a C here, the transcription factor will still bind, although with a slightly lesser affinity than to the one with the most, so the most preferred sequence. So you can think of this as being a spectrum where it definitely prefers binding to certain motifs, certain other sequences, slightly less to some other sort of corrupted instances of those sequences that are determined by its sequence preference. And then, of course, as you go farther away from the preferred sequence, the binding affinity reduces further. So again, like at this position, for example, what this suggests is that it really prefers an A. And if you substitute this A with a G, the transcription factor would probably not bind. So these position-specific preferences are nicely and neatly encoded in what's called this position weight matrix, and we'll talk about that more. So often it's not just one transcription factor that's binding at a regulatory element, but often you'll see binding sites for multiple transcription factors. And often the activity of a regulatory element will increase if you add more transcription factor binding sites, because you can have more molecules of that protein sitting at the same time and sort of bringing about regulatory activity at that element. And just to put this in context, often this regulatory element will be sitting here, there'll be a gene somewhere here, and then physically the transcription factor, so because this is all in 3D space, it's not really a linear, it's not just a 1D object, there'll be a 3-dimensional interaction between the proteins binding in this regulatory element and the gene that's being transcribed. And so having more regulatory elements here can increase the number of protein molecules that you get from a gene that's sort of close by, that's close enough to this regulatory element. So, you know, sites that have multiple instances of the same motif, you can call them as homotypic sites. It's not really that important. But what's important to note is that you don't need to have you don't need to have motifs for the same transcription factor element. In fact, if you look at any arbitrary regulatory element in the genome, it'll be composed of motifs of more than one type of transcription factor. And often what can happen is you can have transcription factors that are specific to that certain cell type. So often a lot of these transcription factors are cell type specific. And so these elements, again, we're talking about livers. So it's quite possible that this transcription factor is highly liver specific, and this site is only bound in the liver. And if you're in any other cell type, this transcription factor will not be expressed and the site will not be bound, right? So this is just the presence of this molecule and the underlying sequence property allows it to activate this regulatory element in a cell type specific manner, right? And so often it's not just one motif, as I said, but it can also be combinations of different motifs. So for example, this regulatory element will only be active when both of these transcription factors, or in fact, all three of these TFs are active at the site, because this site contains elements, sort of motifs for all of these three transcription factors. But even if one or two of them are missing in a specific cell type, you can imagine that this regulatory element will not be active. And so just the sequence here by itself allows us to finally control regulatory activity of certain elements and the genes that are associated with them in a highly cell type specific and sort of combinatorial fashion that's determined by the sequence and the presence of the transcription factors. Right? Any questions? So if you're focusing on a specific regulatory element, we would like to think about how do these motifs interact with each other, and what are sort of the constraints that determine regulatory activity and the strength of this regulatory element. And so this leads to what's called the grammar of spatial grammar of motif combinations, or really the regulatory logic is often also what people call it. But you can imagine that it's not just the presence of these motifs that matter, but also the space between them, right? So sort of if I kept moving them farther apart, let's say I move them like 1,000 bases apart, now these two are really not in physical proximity to each other, and they're really independent of each other, right? But as I keep bringing them closer, you can see a synergistic effect where having one of them also increases the activity more than you would just expect just from the activity of two elements themselves, because now you increase the chances of both proteins coming together, right, at any point in the cell. And often when proteins come together, there's protein-protein interactions that sort of can increase the regulatory activity of the cell. So things like the sequence itself, also the space between the sequences, are really key properties of regulatory sequence that can determine the activity of these regulatory elements, again, in a cell type specific fashion. So how do we even learn these? Before the era of deep learning, how did people even learn these sequence motifs? In fact, a lot of people knew most of these sequences of sequence motifs of most transcription factors even before deep learning was invented, right? So it's important to sort of learn how this was done, why this makes sense, and how you can connect this to the deep learning analyses that we do later. So let's just assume someone gave us the bunch of sequences that this protein bound to, right? There are many experiments to do this. CellX is one of them. It doesn't quite matter, but let's assume someone performs an interesting experiment, and for a specific protein, they tell you that these are the sequences that this protein binds to, right? And these are all aligned, right? So you know that it is binding exactly at these elements at these bases in this longer sequence. And so what you can do is just sort of list these sequences in order. So this is sequence 1, sequence 2, sequence 3, and 4 that are bound on different molecules by this protein. And from this, you can start seeing some patterns emerge, right? You can see that the model almost always prefers a G here, prefers an A here, prefers a T here, an A here again. But at the flanks, again, it's kind of not as strict, right? So here you can see that it prefers not just a C, but also a G at times, prefers an A and a T at the flanks. Right? So from this, again, you can start imagining what the motif of this transcription factor looks like, and I'll of course show that to you. But we can start converting these sequences into what we call a position weight matrix. That's just representing, again, at each position on the x-axis, the preference of this transcription factor for each base, right, A, C, G, and T. So again, as I showed you before, at the second, third, and fourth position, it has a unique preference, right? So you can see at the second position, which is this, it prefers a G, strictly a G, right? And A at the third position, T at the fourth position, and A at the fifth position. But at the first and the last positions, it has an equal preference for two bases, right? For G and C at the first position, and A and T at the final position, right? So this is one way you can sort of convert this list of strings to a mathematical object that then you can use as a model for what this transcription factor prefers binding to, right? And so you can also convert this position weight matrix into a PWM logo. And you can now see a nice correspondence between the sequences that are bound by this transcription factor, how you represent that mathematically, and also how you visualize that, right? Again, it's showing you that the model, that the transcription factor prefers this GATA motif at the second, third, and fourth position, but has soft preferences at the end, right? And so now, given a new sequence, we can start thinking about whether, so given a sequence we haven't seen before, and we don't know if a transcription factor binds there or not, we can start thinking about, just by looking at this motif, whether that sequence will likely be bound by the transcription factor or not, right? And I'll show you how we can do that. So let's say we're given a sequence that has a specific, so we can just convert that PWM matrix, this PWM matrix, into what we call a position specific scoring matrix. It's not that relevant, but you can just imagine as this as performing some division and then a log transformation. But again, this matrix again just contains the same information. You can see it's got at the second position a high score for G, a high score for A at the third position, the same scores for G and C at the first position, and so on. So just a transformation that just makes it a bit easier for us to score a given input sequence, right? So there's just a one-to-one correspondence between those matrices. Now, we'll keep working with the PSSM matrix, but it's all right if it's a bit confusing. You can just think of it as containing the same information as the PWM matrix. And let's say we're given a new sequence that looks like this, and we don't know if it's bound by this GATA transcription. In fact, it's a real transcription factor, and its name is GATA because it likes binding to the GATA sequence. So clearly, genomicists are very creative people, as you can see, right? So given this new sequence, what we can start doing is look at this matrix and then convolve it, right? And really, this is central to the idea of convolution neural networks, and it'll come up again and again. But the idea is if you just drag this sequence across the entire, this matrix across the entire sequence at each position, you score how the sequence, the length 6 sequence beginning at every position matches this matrix. You'll get a value basically for each position. That'll tell you how good the sequence starting from that position matches this matrix. So let's see how we can do that. So what we'll do is, again, you can see that the sequence here, starting at the first position is a GCATTA, right? So you can just start adding up the numbers in this matrix to get a score that tells you the match score of this sequence, so this GCATTA sequence with this GATA matrix, right? So you can see at the G position, so as you remember, it had a preference for GATA here. Clearly there's a C here, so this is not going to work out really well at this side. It doesn't have an A and a T here, but it doesn't have an A here, right? So if you sum up these values, you get a score of about 2, and you start repeating this process where you slide this matrix and you get another score. So you can see that the score at the second position, so for the CATTAC subsequence is lower than the score for the first sequence. And you can also kind of see why, because it's really not a great match. And you can repeat this process as you slide this matrix across the entire sequence, and that'll give you scores at starting at every position in the input sequence. You can see a good dynamic range here. There are clearly sites that have very low scores, some sites that have sort of mid scores, slightly above 0. And you can see this site has a really strong score of 16, and you can see that's happening because it does in fact contain the GATA site, right? With a C here and an A here, right? And so often what we can do is perform some kind of thresholding. And if you're familiar with nonlinear activation functions like ReLU, this is an example of that. What we're just doing is setting all values that are lower than 0 to 0. And for all the values that are greater than 0, we keep them as is, right? And so if you perform this kind of thresholding, which is reminiscent of it is a nonlinear operation because it's an element-wise nonlinear operation because clearly it's not linear, right? Because it is linear for numbers that are positive, but numbers that are below 0 get clamped to 0 here, right? So just going bottom up, you can see that once you get these numbers, we can threshold them. And then this suggests that perhaps there are 2 positions that the GATA motif might prefer. Of course, one of them is preferred much more than the other. As I told you, it's a spectrum. Yeah.
Yeah. So what is it that you feed into the scoring? I mean, you're feeding the genetic sequence.
Right. Exactly. Yeah.
But I'm still not clear. How do you get the weights?
Exactly. Right. So what I'm telling you now is let's assume we have the weights. Someone performed this experiment that I told you about before, right? And they figured out that for the specific transcription factor, this is the sites that it binds to from another experiment. So you can imagine that they went to the lab and did some tedious experiments, not on human genetic sequences, but on some synthetic sequences. And they're able to figure out that these are the sequences that this transcription factor binds to.
So you're feeding in that information in order
yes,
to
for now, right? So exactly. So that's the distinct. That's how it was done in the past, but it helps you build some insight. Later, actually, the motifs will be learned as part of the training process. We won't pre-specify them, right? All right. Yeah.
Next slide.
Yeah.
So I just want to know, how do you calculate the denominator?
Yeah, so usually it's just set to something like 0.25. You give an equal probability to each base. So you're saying that if your random assumption was that each base is equally likely, what is the fold change? What is the ratio compared to random probability, right? So it'll be divided by something like 0.25, and then we take the log of that.
But if it is like organism specific and all, and it does not have that.
Yeah, exactly. So this is a great question. So this background probabilities can also come from the background probability in an organism. Certain organisms have genomes that are more GC rich than others, right? And so in that case, the probability for a G can be based on sort of the base frequency of G in that genome. For humans, it comes out to about 0.25, about 0.3 to 0.5.
Thank you.
All right, great. Thanks. So great questions. And, once we threshold, now given this new genome sequence is actually from our genome, we can now get a sense for whether this transcription factor, again, this is a very simple model. It's not the most accurate model in the world, but it's nice, helps us build some intuitive understanding of what's going on and how these transcription factors bind sequences. So we can start making this model more complicated using, a neural network. Again, takes in DNA sequence, which we convert into a one-hot representation. I'm not sure if you guys are familiar with that. Very simple, right? Clearly, you can't feed in a string to a machine learning algorithm. You have to give it a matrix of values, right? And the way you convert that is using this thing that's called a one-hot encoding, the C. So you basically have at every position a vector of length 4, because we have 4 alphabets in our vocabulary, right? In English, of course, your vocabulary can be much larger. And again, in proteins, your vocabulary will be of size 20, right? Because there's 20 possible amino acids. Here in DNA land, we just have 4 possibilities. And so you code that with a 1 at every position for the base that's present there, right? Straightforward. And then we can have these sort of, the matrix that I talked to you about before, being a pattern detector at each of these positions that really scans across the entire sequence. So again, really what's just happening is that each of these pattern detectors is just this matrix. As I mentioned, it was pre-specified before, but once we're training models, these can be initialized to random weights, and we can learn them in a data-driven fashion. And so these weight matrices can compute just the same way in the previous slide, a score that matches that computes how well the subsequence matches with this matrix. Right? And again, you can kind of do that across all positions. And also, as I mentioned, you have this sort of ReLU nonlinear unit that you can use to the output of the math scores in each of these sequences, right? So you don't necessarily need to have one detector. Now we're starting to think about models where there's maybe not just one motif that's relevant, but perhaps there's 3 motifs that are relevant. This can often happen because the overall activity, as I mentioned, at a site isn't necessarily dictated by the presence of just one transcription factor, but multiple transcription factors, each of which can have unique motifs. And so you want to represent and you want to have the ability to learn these different motif detectors. And so instead of just having one filter that you learn, so just one matrix, you can in fact learn multiple of them, right? And then again, you scan these multiple, so just showing you one, but there's actually multiple motif matrices, each having a different set of weights. You can just scan those across the sequence. And as I showed you before, you get values at each position corresponding to the match between that subsequence and each of those motifs that are being learned. Right? And you can sort of tag in more. So, if you're thinking about this in a neural net, using a neural net way of thinking, you can tack on sort of more layers that help you learn combinations of these motifs. And so typically these are fully connected layers that you can learn on top of the outputs from the first convolution layer. And you can combine them in various ways to get sort of one final output where you have this fully connected layer in the end. And that can help you figure out whether this transcription factor is bound, is this region accessible, what's the activity even of this region in this specific cell type. So that's kind of a high level way in which we'll set up the neural network. One operation that we use that will come up in the tutorial is something that's called max pooling, right? So again, imagine that we are learning different filters and we're able to figure out how well each subsequence maps to each of the filters that we have learned, and we get a value for every position, right? So these are the 5 values we get here. Now, a max pooling operation, what it does is that it looks at the neurons for a fixed width. So in this case, it looks at the neurons at these 3 positions and takes the max value of the outputs of those. So just maybe going back to these slides, let's think about what a max pool will be doing here. So let's say you have, you compute these score matches and then you threshold them. What a max pool is telling you is that I don't really need to know these values at every position. Just tell me within a block of say 10 positions, like between or 5 positions, is there a motif, right? So all it'll do is perhaps like for these 5 positions, it'll just take the max, which is 2 here, right? So you're losing some positional information, right? You're losing some resolution. But often if your task is just to be able to determine whether the transcription factor is bound there, you don't really need to know where it's bound, but more about like, is it really bound at any position, right? And so what the max pool operation will do is take the max of say these 5 numbers, which is 2, and then output a 2 here, and then they take the max of these next 5 numbers, which is 16, right? So that's what a max pool operation does. Why it's nice is that it starts compressing your neural network. It reduces the number of positions that you have after every operation of max pooling. And so that becomes more parameter efficient. And it's just typically that's also an idea that's heavily utilized in computer vision, as you guys might have seen before. And so just combining sort of this convolution scanning, the max pooling operation, and some fully connected layers and non-linearities, you're able to come up with this sort of final neural network that has the ability to really learn de novo motifs that are relevant for any given sequence. And so, this is an example of a single task model. So often these neural networks come in multiple flavors. And in a single task model, you're just predicting one output from a given input sequence. For example, is this transcription factor bound given this sort of input sequence? But often what you can also do is multitask, where instead of having just one output, you can have multiple outputs. An example of that is trying to predict whether 2 transcription factors are bound at this given sequence. So you have individual predictions for the first transcription factor and the second transcription factor. And often the model can learn the rules that govern the binding of both of these transcription factors and learn the relevant motifs for both of them. And often, as I told you, there can also be shared logic between the binding of 2 transcription factors. Perhaps 2 transcription factors have their own unique motifs, but also have a shared motif from a third transcription factor that they both like binding to, right? So we don't have to feed in any of this knowledge beforehand. It's just that the model learns these in a data-driven fashion. And the nice thing is we can share parameters and solve sort of 2 problems at one using multitasking. In fact, if you look at the latest and sort of the biggest neural networks that are trained in genomics, they have on the order of 5,000 tasks, right? So they not just 1 or 2 tasks, but 5,000 tasks that are looking at the binding of different transcription factors in different cell types, looking at overall activity of regulatory elements, again, in different cell types. You can think about, you can imagine how big a combinatorial space this is, not just computationally, but also experimentally, and to characterize. So I think let's stop here for questions, because I've covered enough to actually start the tutorial part. And since we do have some time left now, we can kind of start jumping into the tutorials and build these models from scratch by hand. But if there's any questions, burning questions, let's get them out the way now. Yeah.
Can I go?
Yeah.
So I just want to know how you calculated those weight scores, like
yeah,
done, how initially it was done, like the SSM scores. Or...
Yeah. Okay, that's a great point. I think I'll just go back to what Anshul was talking about yesterday, which is supervised learning. So these are all randomly initialized, right? You'd actually, when you're training a deep learning model, you don't assume any knowledge typically of motifs, weights, and so on, right? And so you really go into this paradigm of supervised learning where you initialize these sequences randomly, right? And let's say you have a lot of labeled data, right? So you have a lot of labeled data where you have different sequences and their labels, right? Let's say on the order of a few thousand sequences. And here this classification or regression model is a neural network that we've randomly initialized. And if you remember from like yesterday from Anshul's talk, now we're just trying to optimize this neural network. We just, you typically have a loss function that maps the outputs of your model on all of these sequences to the actual labels, right? So again, we have not actually initialized, once we initialize a model, all the weights are random in the beginning. And then we just begin this process of supervised learning. And you'll actually see this in the tutorial today. Typically for binary classification, we use a sort of binary cross entropy loss. If you have more continuous tasks, we can use a squared loss. But I hope this was clear yesterday, because Anshul spent some time on this. But as you go over the process of training these models, right? So you randomly initialize all weights, And in this case, it means randomly initializing not just the weights of the models, but the matrices that learn the PWMs, right? And again, at each step, you make these gradient updates and the model performance increases slightly. But what's happening in the learning process, and we'll see this actually as we train our models today, is that the weights that were randomly initialized in the beginning start converging to relevant motif features, right? And this is just a byproduct of the learning process. It makes sense, right? If the model must learn, if there's some logic that's underlying the sequences, and in fact, it's like specific subsequences and motifs that are driving it, then the model must learn that in order to have a high predictive accuracy, right? And so it's really just a byproduct of the learning process, given enough training data and enough sort of labels. Yeah, so I'm just, this is not relevant. Yeah.
Thank you.
Yeah.
You have discussed a lot you have used non-linear model, non-linear function.
Mhm.
So what I can see in that graph, it's like it is linear, but it is censored at 0, or means it's like you have made all the values below 0 is 0, right?
Correct. Yeah.
So how this means function works?
Yeah, great question. So this is not just specific to deep learning in genomics,
Yeah.
but if you look across any deep learning models today,
Mhm.
they all follow the same sort of principle, which is you start off with some linear operation. So as you can see, this convolution operation is linear, sort of at each position, it's linear.
Yeah.
Then you transform that using a nonlinear function, a very simple nonlinear function here, right? Which is just clamping the values below 0 to 0, right?
Mhm.
And then you repeat this block. So you have another convolution layer, a max pool layer, another nonlinearity. So often you'll have some linear operations, right? Which can be structured, right? Because this convolution has some structure to it. It's not just a linear combination of all the inputs, but it's rather position dependent, right? So each output only depends on inputs in a certain subsequence in the input. And so this recipe is really common. So you'll have, even with, if you're familiar with fully connected layers, that's just a matrix multiplication, right? You take in some input, you multiply that by a matrix, again, which is a linear operation. And then you perform, you apply nonlinearity at the end, a position-wise nonlinearity. And then again, you have another matrix multiplication and nonlinearity, right? So in this process, what ends up happening is you have multiple blocks of these linear, nonlinear operations, right? And so this actually allows the model to learn any. So theoretically, people can show that given enough blocks of this kind, these models can learn any nonlinear function.
Okay, so what I can see, it's a combination of uniform plus linear means, right?
Yes.
So, and you are saying this is non-linear means.
Yes, exactly right.
Uniform plus linear equal to non-linear according to this ReLU function, right?
So this entire thing is nonlinear, right? Till here it's linear, and then it becomes nonlinear. And once it's nonlinear, you can't make it linear again. Anything downstream of that, if you look at the output, will be a nonlinear function of the input and the weights.
Okay.
If I'm answering your understanding your question, basically once you threshold it, it's nonlinear. You're cut off.
Yeah.
Okay.
Sorry, actually, yeah, there's one question.
Hi. I just had a confusion in the sense that in the scoring weights, these are the weights that you randomly initialized and then you learned through the process, right? Is it that, or is it the PSSM matrix thing? I just got a bit confused.
So let me just clear the confusion. So the first example I showed you was assuming we know the PSSM matrix, how do we score an input sequence, right? In general, we don't know this. And so when we're training, so this is how it was done traditionally, when these are like simple models that we used in the past, where using some other experiment, you're able to figure out what the PWM PSSM of a transcription factor looks like. And then given this matrix, you can score any input sequence, right? That's kind of the traditional way of doing it. But the more modern sort of deep learning inspired method is once you have a lot of supervised data, you build up this neural network, right? That looks something like this. You initialize these weights randomly. So these models don't actually know anything about specific motifs. And once you learn these models using sort of your loss function, your gradient updates, over time, these PSSMs converge to the PSSM for relevant motifs. Yeah. There's more questions? Yeah.
Sir, can you please shed a little bit light upon the data that you're feeding into the model? Like here
okay.
that you're feeding the input sequence. So here we are feeding only the like large number of sequences, or we are feeding the motifs.
Yeah, so just raw sequences. So, I don't think we have a lot of time to cover the experimental modalities, but this is what the setup really looks like. In general, this is what the setup looks like. You have a lot of labeled data. So it's just sequences, right? Sequences that can be 100 bases long, can be 500 bases long. You can perform certain, let's say you're focusing in a specific cell type at a specific transcription factor and trying to figure out where it binds, right? So people perform experiments where they really pull down the regions that are bound by that transcription factor, right? So you really just pull down those parts of the DNA, you get rid of the transcription factor, and then you sequence the reads, right? So you basically sequence the genome, the parts of the genome that are bound by that transcription factor. And those regions that are bound end up being a positive example because now you know that this sequence was bound, but you don't know where exactly so the sequence can be pretty long, it can be 100 or even 500 bases long. You don't exactly know where it is within this longer sequence because as I told you, these motifs are usually pretty short, like 10 bases long, right? So actually you don't know where it is within those longer sequences that it's bound. It can be at the edge, it can be in the middle, it can be at the end. So we don't actually know this ourselves, like if you don't, without any pre-existing knowledge. And so here sort of it's highlighted that, let's say this transcription factor prefers binding this 80-rich sequence, it can really be at different positions, but we don't know this a priori, and we don't provide that information to these materials. So it's just chunks of the genome that are bound by the transcription factor, but we don't know exactly like what subsequence of that longer sequence is bound. That's generally the paradigm that we work with. That's the experimental outputs that we typically work with. And so you can have labels associated with. You have a label associated with the sequence, but not necessarily with each base in that sequence.
In the slide where you showed the threshold, this thresholding score, so choosing the maximum score, like there it was 16, even if you don't go for maximum pooling.
Yeah.
So let's say you are picking the 16. So choosing the maximum score may result into losing the information about the conserved region or repeatable region also, right? Because that's just a sum that is not telling you.
Yeah. So again, this really depends on the task at hand. If you just want to come up with a binary prediction of, is this transcription factor binding anywhere in the sequence? You don't need to know exactly where it's binding. You can just sort of pull information so that so long as it's binding at least one position, once you max that, yes, you lose position information, but you know that, okay, there's at least one site that is being bound, right? So you're right, it loses some positional information. Sometimes, and there are tasks that we're interested in where we don't want to lose this positional information. In fact, we do use in our lab architectures that don't have max pooling layers at all. So you can keep all the positional information propagate,
But I guess repeating this calculation again and again will somewhere ensure that the position, the conserved position, you will get those conserved positions, I guess, right?
Yes, exactly right. What you're saying is, if there is a good match for that motif, like this GATA here, the 16 will keep propagating up and you will know that there is a binding site. You might not at the end know where it is, but you know it is there, it exists. So again, this is very task dependent and depends on what you want to learn and what you want your model to learn.
Okay, thank you.
One question from me. So the same sequence can be bound in one cell line and not bound in another cell line, depending on DNA accessibility. And so does that go as an input anywhere into your deep learning?
Great question. So not in this model. Typically, these are sequence models, and you're right. When you're looking at transcription factors, it's not just the sequence that makes it accessible.
Or deep learning network.
Right. So most of our models are sequence only. But we do train on regions that are typically accessible, and then our negative regions can be other accessible regions that are not bound by the TF. But in general, we don't add. There are models that use DNA accessibility as an additional input. For making predictions. Again, it's like it's useful for specific. It depends on the biological question you're trying to answer, and it's relevant. So, for example, if you want to know, there are some proteins that are pioneer factors that can open closed chromatin. In those cases, you want to understand which transcription factors are pioneers versus which ones are not. There, you can add the DNA as an additional input and be able to figure out which TFs even open, even though there's closed chromatin.
Yeah, thank you. So I have a basic question about the application of CNNs to sequences. I'm a great fan of this work, but also more recently, there's been a few papers, NeurIPS, which talk about why tree-based models still outperform deep learning models wherever we have discrete sort of data, because deep learning models are based upon smooth gradients. And wherever we have bends and discrete data, sometimes there's non-differentiability. So the models which work with these cuts work better. So a question is there a reason to think that CNN models for DNA sequences, which are unlike CNN models for real world, where there are smooth curves and transitions, gradients, etc., is there a reason to believe that these would still perform as compared to, let's say, the transformer kind of models?
So I guess so is your argument against using deep learning for
no, not deep learning, CNNs.
Yeah, so
especially from the context of the kind of nature of data that we are dealing with. When we don't have a I'm not sure if representation of a genome in a CNN model will have smooth transitions. So I think that's where I'm coming from.
Yeah, so great point. I think the best analogy here is to compare. So DNA sequence is not like a language like English, right? If you imagine an English sentence as I'm speaking, really every word has a certain importance. And if you delete or if you mutate every word or even one word, it can change the meaning of entire sentences. That's typically not the case in DNA, as you said. There's a lot of inactive sequence, and sometimes there's like patches of really active parts. Like, so for example, in this sequence where you have these motifs, that's an interesting part of the sequence. Everything else is kind of dormant and so actually this is more like looking at an image, right? If you expand this to 2D, you can think of that as being, really points of highlight. There's, you can imagine a 2D image and there's like points in that image, and that's predictive. So from an image perspective, I think the convolutional neural network makes sense. However, you're right. I think people have been trying transformers. Transformers have been doing well on DNA sequence data. And so I think the future really is another reason transformers are interesting is because when you start looking at long sequences, like 1 megabase, 1 million bases of DNA, the DNA starts behaving more like a graph at that point, because now you have regulatory elements at different distances, and these sort of interact with each other in interesting combinatorial ways, and those can really be learned better with a graph neural network like a transformer.
Right. So no, it's exactly the point that you said, that when you actually visualize this as an image, you might see points that are highlighted, which is a very non-smooth function.
Yes.
When we are looking, but CNNs by their history have been built for the smooth gradients that operate in the real world images. So I'm just wondering if the application of CNNs on top of transformers based representations is a better way to go, where transformers can give a smoother representations and then you have CNNs to work with. And that's opposite to the other way around, right? So I'm just, and we've done some work in transformers for COVID-19, as I was talking to you about, but haven't explored that. So would appreciate your thoughts.
Yeah, so so even if you think about how it's really pointy in nature, right? These images really have the sharp. That's not really been an issue when we've trained these models, and there's been a lot of benchmarking done against more traditional sort of SVMs, and because often so a lot of these models like SVMs, they lose positional information. And if you think about, yes, there are 2 points that are quite sharp, but also the distance between these points matters, right? And those things are hard to model, especially if you don't know what those points are in the first place. So typically when you're modeling regulatory sequence, as I told you, we don't even know what the underlying feature is. That's actually learned as part of the model. And so things like spacing between these with motifs is also very predictive, is important for making accurate predictions. So while you're right, it's quite pointy, it hasn't been so much of an issue as far as I can tell. And there's been a lot of benchmarking in general. But you're right, for the long range models I was talking about, that taking like 300,000, 3 lakh basically bases as its input, you still can't use a transformer on a sequence of length 300,000, 1 million. And so that the strategy we will have adopted is convolutions, max pools, convolutions, max pools. You do about 10 layers of these, you shrink the sequence down from 1 million to 1,000, and then you do transformers on this sort of compacted sequence. I think that's a good trade-off between using both transformers. using convolutions to sort of reduce the effective sequence length and then transformers to learn all kinds of pairwise interactions. Because I see another point in favor of transformers is that convolution, if you think about it, the prediction at this position does not necessarily depend on the sequence, say 100 bases away in the first layer, right? Perhaps after 20 layers, there'll be a link between bases that are far away. In a transformer, after your first transformer layer, everything is connected to everything else, right? So I think that probably aligns more with, I think the combination of convolutions plus transformers makes the most biological sense.
Thank you so much. Would love to chat more. Thank you.
I think we had some more questions. I guess we're also out of time, so.
Yeah, we have a few minutes left, so either we can have more questions or you have something to.
Okay, we have one.
Yeah, so what about the sites that are physically inaccessible?
Yeah, great question. So sites that are physically inaccessible. So when we often train these models, we'll not talk much about that because that assumes some more biological knowledge, but great question. Typically, when we train these models, we ignore sites that are not accessible. If you're looking at a specific transcription factor, when you train these models, your positive examples will often be regions that are accessible. And then the negative examples, you can choose them from regions that are accessible but not bound by this TF. Or you can also choose regions that are in general not bound by this TF. So I think the point is that if you learn this model and you try to make predictions genome-wide, it'll fail at a lot of positions. A lot of those positions will just be physically inaccessible, and so the transcription factor can't bind there. However, there's a small category or class of transcription factors that are called pioneer transcription factors. And these transcription factors have the ability to bind compacted DNA and open it up, right? So if you've heard of the Yamanaka factors and reprogramming from to stem cells, right? This is something that came up about 15 years back. What happens in that process is that you overexpress these Yamanaka factors. It's like 4 transcription factors that are very potent and linked to stem cell identity. And when you overexpress these factors, they basically recognize their sites on compacted DNA, and then they bring about sort of other cofactors and proteins, and they end up opening those sites. So I think people are still starting to think about how to model those kinds of behaviors, and but I think it's still a area of active research. Thanks.
Yeah, so it is 10:31 now, so we can have a coffee break and we'll rejoin at 11. So thank you, Surag.